All right, good evening. Welcome to the MyCP webinar series, second webinar for 2021, where tonight we have the pleasure of Dr. Michael Kruer talking to us about genetic causes of cerebral palsy and his study that is being launched and that is NIH funded and we think is going to make for some great findings. And I look forward to having him tell you about those. So the agenda for this evening is I want to just give you a quick introduction to the new CP Research Network. I'll explain that in a moment if you're not familiar. And then I'll turn the reins over to Dr. Kruer for his presentation, and then we'll have an open Q&A. Okay, so many of you have probably seen this graphic, but in late January, Michelle Schusterman and I announced that we were merging the CPRN with CP Now. So CP Now is famous for the CP toolkit, and originally Michelle started CP Daily Living, which was a blog that got a large following. And so we are in the process of taking these four websites, merging them together, but we've merged our operations. We're all one 501c3, and this is our new logo representing the Cerebral Palsy Research Network. So our new mission is optimizing the lifelong health and wellness of people with cerebral palsy and their families through high quality research, education, and community programming. So I want to explain a little bit about the network. This is my sort of standard pitch that I've done on other MyCP webinars. The network is a 501c3, not-for-profit organization, and its main effort is it organizes the field of institutions, universities, clinicians, therapists, people interested in research, and the community, patient advocates, to conduct research to improve outcomes for people with CP. In addition to this sort of collaboration effort, we are a data coordinating center, a place where the data comes together for study at the University of Utah in our Department of Population Health Sciences. What the network does is it provides a lot of infrastructure to people interested in research to help facilitate that research. The core thing we provide is a clinical and a community registry. The clinical registry is collecting information about visits to the hospital, either for interventions or for going for annual clinic or for injections or something like that. And the community registry gathers data about people's lived experience with CP while they're not in the clinic, not in the hospital. And then with the merge with CP Now, we have picked up education and well-being programs as a core part of what we do. MyCP is an essential part of that, and MyCP is a web portal that allows you to come and have a little bit more personalized experience about your CP and the opportunity to participate in research. So first and foremost, it is the way in which we give people access to those community registries. In the community registry, you're participating by filling out surveys that you can do either on your computer or on your phone. It also has a private place to have discussions with other members of the community and with clinicians interested in research to share about lived experiences, important research questions, and to collaborate really on research. And then finally, it is a place where we are building out a set of tools to help people manage their experience with CP. The first tool is just a gross motor function classification scale tool. When you sign up for MyCP, it takes you through the process of assessing your GMFCS. And so that is the start of what we think we can build out to be many tools to help with things like transition, with visits to your physician, and to make sure that you've got good information to help guide those discussions. So that is a place we're continuing to invest in tools. And anyone can sign up at mycp.org. So the clinical registry currently has, it actually has just over 5,000, but this report is from when we had 4,485 unique patients. So people come into the hospital, come into the clinic, and we are gathering routine clinical care data that both describes people in terms of demographics, as well as their diagnosis for CP, their limb involvement or CP distribution, what is the etiology, but it also characterizes interventions they have, neurotoxin injections such as botulinum toxin A, Selective dorsal rhizotomies, that's that SDR abbreviation. We have 150 of those. The implantation of an intrathecal baclofen pump. And then we also look at orthopedic surgeries. So we're capturing information about all of these interventions. Oh, sorry, accidentally went ahead one. So the graph here is just to give you an idea of one piece of data. I don't know why this keeps going that way, but I apologize. So this is a graph of the gross motor function classification scale, that Roman numeral 1 through 5, that describes people's ability to ambulate, or if they use a chair, how much head support they have. So that covers the range from GMFCS 1 through 5. And you see this is sort of the distribution of CP, not in the population, but in the hospitals that are participating in the registry. So we're not doing this to surveil the population and see if there are changes. We're doing this to characterize the population that comes into a clinic setting and potentially gets treatment. So that gives you an idea of how the registry is used. So the CP Research Network has several programs now. So this is a picture in the upper left of our investigator committee when we last were able to gather in person. This was in 2019 in Ann Arbor, Michigan. As our host, Dr. Ed Hurwitz there. And the group does both research and quality improvement, which is a way in which you can very rapidly improve outcomes through changing the way you deliver clinical care. We're very focused on community engagement. That's a core pillar of what we do. So this is a shot from our 2017 workshop that was the culmination of our research CP, that we did that to set a patient-centered research agenda for cerebral palsy, and that's now driving what we do for our research. We also have a number of health and well-being programs that we're working on. We're just in the process of launching Mentor in conjunction with a National Center for Health, Physical Activity, and Disability. That's aimed at adults, and we've been working with Shriners to build a nationwide program for kids for participation in physical activity. We have the MyCP program, and then the resources that have been developed originally by CP Now that are part of the CP Research Network. The CP Toolkit, which is great for people with an initial diagnosis, and a well-being guide that's aimed at having parents sort of understand the stresses of having a child with CP and how to take care of oneself. So we'll ask, so where is it? This is our current map of where the clinical network sites are. We have about 29 sites at this stage. The green pins are sites that are actually enrolling patients into the registry now. The yellow pins are ones that are working on the technology to support the registry. It's very deeply integrated with a hospital's electronic medical record system. The red pins are sites that are actually just doing the compliance work of getting an approval to participate in the studies or to share the data with the University of Utah. And then the blue pins are sites that have raised their hand and said, we want to participate. And so they're just starting to go through the process of getting onboarded into the network. So with the blue pins, we would be up at about probably 36 sites. So as I said, we exist to accelerate research in addition to our education and well-being programs. So this is our pipeline, where on the left-hand side, you have the front of the funnel, where what we're trying to do is to get studies created along the research CP agenda, and then we take them through to approval, and then they pursue funding through either private or public funding opportunities, and then they get implemented. So tonight we're going to be talking about Dr. Kruer's study on CP genetics, which has been through this whole process and is now in the implementation phase and the study execution phase. The orange bubbles are some of our quality improvement work. So you see that we're working on dystonia. We're working on adult-centered care, hip surveillance, and we've had one for a while working on reducing infections for intrathecal baclofen pumps. So this is the idea of how we are creating a lot of research. And then you can see we're working on a lot of publications. We have 3 publications out. And then we try to take this evidence base and then push it back into the front of the network. So we're always delivering evidence-based care, improving outcomes for the people that come to CP Research Network clinical centers. So that's the network. During the Q&A session, if people have questions, I can answer more questions. But for now, I'd like to turn it over to the man of the hour and the storyteller of the day, Dr. Michael Kruer, who is the director of the Pediatric Movement Disorders Program at both Barrow Neurological Institute and Phoenix Children's. And he is also an associate professor at the University of Arizona's College of Medicine. And he is a site PI for Phoenix Children's for the CP Research Network. And he has the feather of having captured our first public funding from NIH. And it's quite a significant study that he is just now launching. So we've arrived at this hour, and I am going to turn it over to Dr. Kruer. I will stop sharing so you can take the screen share.
All right, thanks so much, Paul, and good evening, everyone. It's really a great pleasure to be able to share this this research study with you all. This has been something that has been several years in the making, and we're very excited about not only where the application of genetics is to cerebral palsy overall, but for the launch of our CP Research Network study that I'll be telling you about at the end. The title of this presentation is Why Genetics Matter, and hopefully I can convince you by the end of this talk that genetics do matter for CP. So this group is really no stranger to the idea that cerebral palsy represents one of the most common neurodevelopmental disorders, and really is the cardinal condition that leads to physical disability, particularly in children, but also in adults. And so given my background in developmental pediatrics, I'm used to thinking about development in terms of these different streams or puzzle pieces. But I think it's worth mentioning that all of these streams of development are important. And although the work that we're doing is focused on CP, we think that there are quite a number of overlaps for other aspects of development, and in particular for other neurodevelopmental disorders. And so we'll talk more about that during this presentation. But I think that what I'd like to start off with is the idea that rare diseases or rare disorders are actually collectively common. And so I think that what you can see here is 3 hands, each of them very different, but yet very, very similar. All of them are hands, and yet each of them are unique. And I think that we're starting to think about disorders like CP in this way. So these 3 hands together make up the schematic for the logo for the recognition of rare disorders. And interestingly, what we're starting to find out is that many of these common neurodevelopmental disorders, whether it be autism, CP, or another condition, there are many roads to these conditions, and that many neurodevelopmental disorders are actually collections of individually rare conditions. So it might be uncommon for an individual to have CP because of a specific reason, but collectively as a whole, CP is thought to currently affect around 1 in 323 individuals. And so when we think about CP, I think it's important to recognize that we really, despite the efforts of many, many talented physicians and researchers, have not had new treatments for CP in some time. And so Paul did a great job of outlining the structure of the CP Research Network. And one of the jobs of the network is really to try to advance the needle for the care of people with cerebral palsy throughout the lifespan. Now, that effort really represents a culmination of both short-term and long-term goals, like many portfolios. And so I think it's important to recognize that in the short term, we're still trying to understand how to best use the tool at hand, the tools that we have at hand, whether that be medications, some of the surgical interventions that Paul was mentioning, or even therapies for individuals with CP. And this represents a really critically important goal of the CP Research Network. As Paul mentioned, we think that this can have a high impact in the near term if we learn to best utilize the tools that we have. However, I think that the genetics study represents a bit of the other side of the coin. And this represents a look towards the long term and a recognition of the fact that we also need more in the pipeline. We need to be thinking about long-term solutions for individuals with CP. And through the lens of genetics, as well as other up-and-coming tools, we think that we can start to repurpose some of the existing medications that might be out there, and even contribute to the development of entirely new therapies, whether those be new medications, new devices, or even things like gene therapies or CRISPR-based treatments, which some of you may have heard of. So I do think it's important to recognize that at this point in time, though, most treatments for CP are really focused on reducing symptoms, not the fundamental problem. And as a neurodevelopmental disorder, I think we've long had an awareness that CP has a tendency to cause brain development to take a wrong turn at certain points, and that this can fundamentally change motor and physical development. Nevertheless, despite this recognition, intervening effectively remains a challenge. And I think that this is one of the reasons that the treatments that we have at this point in time really are focused on symptom reduction. But if you compare CP to epilepsy, another common neurodevelopmental condition and one that overlaps with CP for almost half of individuals, controlling seizures in epilepsy is incredibly difficult many times and can be very complex. But in some ways, CP can be even more complex. Because, for example, in epilepsy care, the goal is to stop the seizures, whereas in CP care, what we're trying to do is not only reduce unwanted muscle tone or take away involuntary or abnormal movements, but we want to fundamentally improve function and hopefully intervene at a point that will have a lasting impact on an individual's life. And so many of you might be asking, well, why genetics? And the reason is simple, but yet more complex than I think we have ever realized before. And that's that genetics can provide an important stepping off point. It really can provide a roadmap for research by helping us to hone in on what the fundamental problem is and where development in many cases may start to go awry. Now, we don't think that genetic insights will necessarily apply evenly across the board to all individuals with CP, but yet we think for the community as a whole, the genetic knowledge is going to really shine new light onto pathways and processes that are important. By better understanding normal brain development, we think we'll also gain a new perspective on the missteps in brain development that might occur in CP. So one of the questions I commonly get is, well, how important is genetics to cerebral palsy? And I used to have a diagram here where I took a stab at this question, but I've replaced it with just a big question mark, and that's purposeful. Because I think that as time goes on, I recognize more and more that we simply don't have a good answer to this question. Current estimates range anywhere from 15% to 40% of CP might actually be related to genetic changes. But I think that this really deserves further study. And this is actually something that we would love to look at further through the lens of the CP Research Network. So many of you might be asking, okay, well, genetics, what are we talking about exactly? And when I say genetics, I'm fundamentally meaning the study of genomes. And so sometimes genetics is also referred to as genomics. And the reason for that is that all of us have a fundamental blueprint that tells our bodies how to grow and how to function. That blueprint is a cookbook of sorts that comprises many, many different recipes that contain the instructions that are needed, again, for our bodies to grow and to function. So that recipe book, if you will, is comprised of many, many individual recipes. And each recipe does something different in our bodies. Some might be important for determining our height, our eye color, how our heart functions. But it turns out that thousands of these recipes are actually necessary for our bodies to build and develop a human brain. There are roughly 20,000 different genes in the human genome, and a large proportion of them are actually important for normal brain development and function. Now, each of those recipes or those genes is written in a code of DNA that fundamentally comprises the alphabet that's used to inscribe that particular recipe. And the net result of this is that we ultimately want to use that recipe in order to have something happen. So for example, if you're talking about a chocolate cake, you could imagine a scenario where if there was some type of typo or misspelling in the genetic code or in the DNA that comprises the recipe for that cake, that could affect the way the cake turns out. And perhaps not too surprisingly, if there's a misprint in your recipe book and you don't know if your recipe requires 2 eggs or 6 eggs for this chocolate cake, that could affect the way this cake turns out. And so these mutations can sometimes be just single letters. Sometimes it can be very remarkable that a single misspelling of one of the DNA letters out of 3 billion can be the difference between health and disease. In other cases, sometimes entire pages are torn out of the genome or the cookbook, or it can even be duplicated, which can also affect the way that these recipes turn out. So when we're talking about genetics, when applying it to CP, I think it's important to recognize that early studies were really asking whether genetics represented an important risk factor. That is, do genetic changes then, for example, increase a person's risk for some type of factor that would then cause CP? And we still think that that's a very, very important question and something that we're going after with our research. But I think at the initial stages, what we're doing is we're really focusing on genetic changes that have a major effect on function. And so in this way, if you think about the difference between health and a disease or a disorder condition like cerebral palsy, if there's a line, so to speak, between the two, where a normal gene without a mutation might lead to healthy functioning of that particular gene, what we're seeing so far in our research, in our research group as well as many others around the world, is that a mutation in that gene can actually tip the scales. And sometimes just one mutation can be enough to tip the scales from typical brain development into CP. And so as far as our initial studies, they're focused on these large effect size genetic changes that fundamentally change the way the recipe turns out. So how does this relate to CP? Well, you know, I think there are a number of different ways that this not only helps to advance the field of cerebral palsy and our understanding of the neuroscience of CP, but we also believe that research is fundamentally good medicine. This is what helps us as physicians and therapists and providers do more and more for our patients each day, rather than continuing to do the same things all the time. And this is one of the fundamental principles that drives the CP Research Network. Nevertheless, I'll be very honest with you, we still have an awful lot to learn. So I'm sometimes asked if I can tell if someone that I'm meeting in the clinic or the hospital has a genetic cause for their CP. And at this point in time, I can't. I can't tell that just by looking at them or talking with them or examining them. I think that what we do know is telling us that the lack of risk factors. So for example, if that child or individual was not born preterm, if they did not have any lack of oxygen, if there was no infection before or around the time of delivery, if there was no stroke or bleeding in the brain, the absence of some of those typical risk factors can be an important clue and might suggest that that individual could have a genetic reason for his or her CP. Interestingly, though, we're also finding that for some individuals that have those well-known environmental risk factors, they still will have a what we think is a high impact genetic change that might also be contributing to their condition. So we still have a lot to learn, and this is the subject of ongoing research. Some of you might be asking, well, why does it matter? And again, I think that's a fundamentally important question. As I've spoken with Paul many times, as a researcher, it's very important to me that the work that we're doing, whether it be in the clinic, in the hospital, or the laboratory, really is something that's important and of substance to people with CP and their families. We do think that identifying new genetic causes of CP might actually shed some light on the biology of cerebral palsy and how these genetic changes might actually lead to changes in the brain that in turn alter development. But another exciting aspect of this work is we're starting to find that there may be immediate clinical implications for people, meaning that sometimes for an individual for whom we identify a genetic explanation for their CP, this can actually impact aspects of their diagnosis. It can help avoid preventable complications. And perhaps most excitingly, this is even starting to lead us to new forms of treatment. I think there are also some very potentially important human implications to this, as I've known a number of parents that have that nagging doubt, if only, and maybe if not for blank, perhaps my child wouldn't have CP. Very seldom is that the case, but we do find that sometimes with our genetic discoveries, we are able to put people's minds at ease because they're able to recognize what the fundamental causes of their child, their family members' CP might be for the first time. So I've hinted around about this, but I did want to share some of our latest findings, and this is boiling down years of work through 5 continents around the world, and researchers, families, and clinicians from many different places. But we recently, just in October 2020, published this paper in Nature Genetics, which really represented the first firm statistical evidence that genetic mutations, in fact, do lead to CP, and that this is more often than we would expect by chance. So this really was the first paper, I think, that conclusively demonstrated that genetic mutations represent an important cause for CP. So as part of that discovery, we did identify several new CP genes. And what's been quite interesting is that there's not just one gene or a couple of genes that can pick up a mutation and lead to CP. But in fact, our current estimates, based upon some modeling that we've done, suggests that actually hundreds of different CP genes exist. And many of these genes are genes that all of us have and are important for normal brain development. But if these genes pick up just one typo or misspelling, again, that can affect the way the recipe turns out. And what we're now identifying is that although it's been well known for some time that lack of oxygen, strokes, bleeding in the brain, or inflammation can be very important and contribute to a large number of CP cases, for other individuals, there have been new pathways that we've identified. And these pathways actually affect the way that brain cells very early on will reach out, form these elaborate trees, and connect up with one another. And you can see that among a bunch of neurons in a dish in my laboratory. And then what ultimately happens is that these handful of neurons in the human brain under normal developmental conditions become billions of neurons. And those billions of neurons form a dizzying web of connections that you can see here through an advanced MRI technique. And what we do know is that for many people with CP, if not most, there are abnormalities of the way that those connections are actually physically structured. And even for those for whom we have a difficult time showing that there are physical differences in the way the brain is connected up and wired, we're now able to identify functional differences in the way that different areas of the brain will light up and communicate with one another. And we think that that's one of the fundamental issues that's leading to motor dysfunction and to CP for many people. So we think that this is important to get out there, but there's been a bit of a debate in the medical community as to whether if you identify a genetic mutation, that person, quote, really has CP. And I think that this has been hotly debated actually for the last few years, but a consensus is really starting to emerge that the diagnosis of CP is independent of cause, meaning that CP, like epilepsy, like autism, like many other neurodevelopmental disorders, is a diagnosis that's based upon a person's clinical symptoms and presentation. And there are many different causes or many roads that will lead to CP. And so this is something that's been the subject of a recent consensus statement. But I think that rare disorders organizations, and if you remember in the beginning of the talk, I was talking about rare disorders. Rare disorders organizations are starting to recognize this more and more. So for example, this particular rare disorders organization, this gene and mutations in this gene so far represent the most common genetic form of CP. And so we're starting to find very interesting overlaps between cerebral palsy and the rare disease community. But I think for a lot of families that we talk to, what they'll tell us is that they don't want to choose between which community they belong to, and they don't think they should have to choose between the communities. They find a lot of value, for example, in perhaps finding other families that are dealing with the same rare mutation in a gene, but they also find a tremendous sense of identity and community in connecting with others, for example, in the CP community. So some of our latest findings that at this point are currently unpublished are certainly exciting to me as a practicing physician. And I think that just to give you a little bit of a sneak peek, we're currently able to identify a genetic cause for more and more of the individuals that undergo the large-scale advanced genetic sequencing that we're doing today, called whole exome or sometimes whole genome sequencing. And so because of this, new genes that cause cerebral palsy are actually being identified at a pretty steady clip on a regular basis. And so that number is growing over time. But as I hinted at earlier, we're starting to find more and more examples on an individual basis of people who undergo sequencing. And we're able to find a genetic explanation for their CP. Now, in many cases, I'll be honest, at this point in time, that does not fundamentally change how we treat them. But for a small but growing minority, we are starting to change the way that we treat them. And we're actually beginning to repurpose some existing medications. So for example, very carefully using techniques that have been designed for rare conditions, applying a new treatment, or as I mentioned earlier, avoiding potentially dangerous risk factors. And we just think that this number, this proportion, is going to steadily grow over time. This is starting to have implications for diagnosis. And so this is a practice parameter for one of the largest groups of neurologists in the world. And this is something that during my own training, I was taught to refer to when thinking about trying to understand the causes for an individual, particularly a child's cerebral palsy. However, this was last looked at about 15 years ago. And quite honestly, things have changed. And so, for example, this practice parameter indicates that there really is not a role for genetic testing in CP. And I think that that's fundamentally changed at this point in time. The prevention is a particularly important aspect and something that I don't want to underemphasize, but you've all heard the saying, an ounce of prevention is worth a pound of cure. What we're really trying to do here is fundamentally prevent problems before they start. And with prevention, in most cases, we don't have the ability to prevent an individual's CP at this point in time. But for example, if we know that a particular genetic cause of CP not only leads to CP, but could perhaps affect kidney function, in some cases, we can use that knowledge and we can head off and prevent complications before they ever arise. And this is something that I think is very exciting as well. I briefly mentioned treatment. I think that this can take several different forms. So the way that genetics can inform treatment can be one of these four different manners. Number one, I think that genetics can sometimes give us new insights. So instead of treating every case of CP or every subtype of CP as the same, if we're armed with new genetic knowledge, that sometimes is allowing doctors to reach out for the best treatments first, rather than finding it through a series of trial and error steps, as many of you are familiar with. In other cases, that's allowing us to repurpose existing treatments, as I mentioned, using non-CP medicines for CP treatment off-label. In other cases, this is starting to allow us to better predict responses to surgeries. Deep brain stimulation is an example that I'm particularly interested in a potentially very transformative surgery, but something, an intervention that has a wide range of responses. So the ability to predict who is a good candidate versus who should go down a different road would be very powerful. And then finally, we think that these new genetic insights are ultimately going to allow us to understand where development takes a misstep and really focus treatment development around trying to prevent or correct that. So I mentioned this possibility of repurposing things. This is something that's really starting to gain traction in my own field of neurology, particularly in child neurology and in reference to neurodevelopmental disorders. As it's starting to be realized that the more insights we have about what's fundamentally causing a child's symptoms, that sometimes allows us to do innovative, personalized treatments that in the past have not been available. And so, of course, this needs to be done carefully and responsibly. It needs to involve all the right ethical oversight and some very frank discussions with the patient's families. But guidelines for how to do this and these trials and individual patients are really kind of at the border or the boundary between research and cutting edge clinical medicine. But this is something that we're starting to do more and more. And so this is impacting a small number of patients at this point in time. But as I said before, we expect this number to grow. So with that, I'd like to tell you about the study that Paul mentioned. This is a study funded by the National Institutes of Health, and it really is fundamentally a cerebral palsy study. It's something that was born out of the support and the framework of the CP Research Network. This study is being jointly spearheaded by our group in Phoenix, as well as the University of Utah group. Fundamentally, we're looking to enroll 500 families. So when we're looking at genetic causes of CP, we want to not only look at the individual with CP, but we also want to look at his or her parents. And the reason is because, for example, if both parents are healthy and don't have CP, we can ask the question, well, how is that child's DNA different than their parents? And sometimes looking at those differences can be the first clues as to what differences exist and whether they might actually be responsible for CP in that person. So the study is focused on families, and we're looking to analyze their genomes and identify genetic mutations that lead to CP. And really, the study is comprised of two arms. One that uses saliva samples to collect DNA, and then to use powerful genomic techniques to sequence that DNA and compare it. And the other really harnesses the magic of the CP Research Network. So for those participating in the network and getting their care at network sites, every time an individual with CP is seeing their doctors, just through the course of caring for that patient, are entering information into their electronic records. That electronic information is actually being funneled into the CPRN database. And it's being done in a de-identified manner that keeps personal information protected. But it allows us to learn as we care for patients with CP. And so by coupling that genetic information with that clinical information, we really have a very powerful tool set with which to try to understand genetic contributions to CP. So as part of this process, we anticipate that we will identify known genes that are known to lead to CP when they develop mutations. But we also think that we're going to be in the business of discovery based upon our results so far. And so we anticipate identifying completely new genes that lead to CP and affect early brain development. So for those of you who might be interested, participation is currently by invitation. So because of the design of the study, individuals that participate have to be getting their care at a CPRN center. And so far, we have 5 centers that you can see listed here that are our initial launch group, but we're already talking with several additional centers to have them added to the study as well. As Paul mentioned, in support of Cerebral Palsy Awareness Month, our go live is actually later this month. And so for those individuals that are interested in receiving their care at one of these centers, we'll be reaching out with the invitation to participate in the study. One of the things that the pandemic has really forced us to do is to think about this in a new way. And so the study fundamentally is aimed to make things feasible for families, particularly during these difficult times. And so people that are interested in participating can enroll. They don't have to go in person to the clinic. They don't have to have a lengthy phone call. They can actually enroll directly from their phone, their tablet, or their computer through a fully self-contained consenting application. And here's a little sneak peek of the application itself. And so the initial part of the I consent is designed to just simply share information about the study. And so people that are interested in the study will go through this first part and simply receive a series of informative information slides and be told about the study. After going through that, they'll then have the opportunity to decide if this is something that they're interested in for their family. For those that are, the app allows them to enroll electronically. And then after the family is enrolled, that will then trigger our group in Phoenix to send a sample collection kit directly to people's homes, where in the comfort of their own home, they can provide a sample, which is just simply a saliva sample, much less invasive than even a blood collection. So in conclusion, we think that genetic causes of CP are substantial, and we think we're really just at the tip of the iceberg. We fundamentally believe that genetic findings are going to have major implications for our understanding of CP neurobiology. But we also were very excited about the prospect that these genetic findings can start to impact the care of individuals with CP. And then finally, we invite your family to partner with us and join the genetic causes of CP study. So with that, I'd like to say thanks to all of you. I'd like to briefly show you a video on the next slide, just presenting an overview of our study. But those of you who might be interested in participating, you're welcome to reach out to us at kruerlab@phoenixchildrens.com. You can find information about the study on the CP Research Network website. And you also can just simply use your search engine and look up Kruer Lab and find us that way. All right, so here's our video describing our study.
Michael, you're not getting audio.
Up.
Want me to try and show it from my side?
Yeah, that'd be great, Paul. Thank you.
Sure. Hold on one sec.
I think the audio helps.
We're inviting you to participate in the genetic causes of cerebral palsy study because someone in your family has CP and receives care at a cerebral palsy research network site. CPRN centers are working together to establish gold standards of care for people with CP today. They are also working to improve the care of tomorrow through research and innovation. As many as 1 in 4 people may have a genetic cause for their CP. The Genetic Causes of CP study wants to partner with families like yours to understand how genetic changes may affect brain development and lead to CP. We also want to use genetic discoveries to improve treatments. If your family wants to be a part of this important work, let's get started. The first thing to do is officially sign up for the study by completing this app. The second is to provide a DNA sample for the person with CP and their mother and father. We get DNA from your saliva. All you and your family members need to do is spit in one of the collection tubes that we'll send directly to your home. Then send the package back to us. Having samples from mom, dad, and the person with CP allows us to understand what is different in the genetic code of the person with CP compared to their parents. Your local CPRN center will provide information about your symptoms so we can better interpret your genetic findings. This information will be de-identified and not linkable to you. It will be kept highly secured, encrypted, and confidential per federal regulations, although no data safety plan is completely free of risk. Joining the study is completely voluntary and will not affect your ongoing care. You will not be paid for participating, but there is no cost to enroll. Obtaining your genetic testing result is possible, but requires additional permissions from you. There may or may not be a direct benefit for your family if you participate, but this research could help people with CP around the world.
Right. Thanks so much, Paul.
Yeah, no worries. So are you ready to open it to Q&A, Michael?
Yeah, I'd be happy to take any questions that folks might have.
Okay. Let's start with, there are some questions that have been placed in the chat. I'll read you. The first question is, are you looking specifically at CP patients that don't know whether they have mutations, or ones that know that they do, or both?
So I think that we are interested in trying to get a survey of individuals with CP across the board. And so I think that the initial phase of the study is going to be focused on those that don't necessarily have a known genetic cause already. But outside of that, we're taking all comers, anyone who's interested.
Okay. The next question was, if my child has several risk factors that make a genetic factor unlikely, would you want her data as a sort of control group?
Yeah, so the short answer is yes. I think we're interested, as I just mentioned, in getting a good sense across the board of what genetic contributions might be. And although it's true that so far we're finding the highest hit rate, so to speak, in people that don't have any other explanation for their CP, they're not the only group that we're finding mutations in. Sometimes we're actually being surprised that the genetic mutations are allowing us to look at things in a new way, or sometimes tie a number of different factors together.
Great. And then there was another question that says, I'm a twin. In the hypothetical case of a genetic involvement, might science one day be able to answer why I have CP while my twin does not, or at least who hasn't expressed CP symptoms?
That's a great question, and in fact, if you're interested in research, I would invite you to reach out to us offline. I think that twin studies are a particularly powerful way to try to get at some of these genetic changes, or sometimes what are called epigenetic changes. Interestingly, both genetic and epigenetic changes have been seen in individuals that have CP as part of a twin pair. And sometimes, even though twins are thought to be identical, they're not completely identical 100%. There can be differences at both the genetic and epigenetic level.
Great. Someone asked, do you expect to find advances or adjustments for non-pharmacological treatments?
That's a great question. And I think we're really interested in using genetics as a tool. So I think we have our initial goals as part of this first wave where we're trying to identify causes of CP. But one of the things that we're ultimately hoping to do, as I briefly touched upon, was to use genetics as a way to help predict who might respond to given treatments. And that includes medications, surgeries, but also other types of modalities, including therapies and other interventions.
I'm going to open the floor to questions. I'm going to ask you to answer one question that I heard you answer the other night that I thought was interesting, that if your child doesn't have the ability to spit readily, how would you be able to collect the saliva?
Yeah. So turns out we've planned for that possibility, and some of the saliva collection tubes that we'll send for the individual with CP, they are specially adapted so that they essentially contain a sponge. You can just simply place it in a person's cheek on the side, and it will passively absorb the saliva, and then you can just press it against the tube, and then it collects the sample that way.
That's great. Good thinking. Okay, I want to open the floor to questions. You should be able to unmute yourself from the sort of participant panel, or from your, not from the participant panel, but from your little control panel. If you have a question, feel free to ask Dr. Kruer. If you don't have a question, I don't know if you can see me, but I'm going to show you. This is the shirt. This is one of the 3 or 4 shirts that's available along with a mug on Bonfire. We're posting about it regularly. If you want to support the CP Research Network and CP Awareness Month, go out and get yourself a t-shirt. Sorry, got to advertise. Thank you, Nicole. I'm glad you like it. I like it. I just opened mine up today. So I haven't received my green ones yet, but okay. Any questions for Dr. Kruer?
One additional point that I'll make is that a question that often comes up is whether there's an age limit to participate. And the answer is no. So both children and adults with CP are eligible. However, just because of the nature of the genetic studies that we have to do, at this point in time, we do need to have both parents, both biological parents, available to participate. So sometimes for adults, that can make things a little bit more challenging, but we're hoping to open this up to a broad representation from the CP community.
Yeah, and I would also point out that since you have to be seen at a CP Research Network center, only some of our centers, a number of our centers have adults that are 18 to 21 or 23, but if you're more adult than that, you would need to be at a center like Nationwide Children's Hospital, where they see people throughout the lifespan. And hopefully as we add more centers, we'll get more lifespan centers, so there'll be more possibilities to get a mix of adults into the study. Well, it's late on the East Coast. I know that Dr. Kruer will be on my time zone soon when we get to daylight savings, but I want to thank you so much. I've really seen this presentation as you published your paper really share so much more detail about the importance of genetic studies in CP, and it's really quite exciting. So congratulations on getting to this point. I really look forward to launching the study later this month, and thank you, Dr. Kruer, and go CP Awareness.
Go CP Awareness.
All right, thank you, everyone.
Have a great night, everyone.
All right, bye-bye.